Today I'm going to be talking to you about something a little bit different, a technology called Ankyrons. So the first thing I'd like just to explain a little bit is maybe a challenge that many of us will be familiar with in a lot of detail, and some of us this might be kind of a little bit new. But particularly in the context of infectious disease and pathogen outbreaks, one of the major challenges that we really face as a community of scientists is the generation of large panels of new reagents to be able to really understand the biology and potentially even diagnostics of these sorts of new pathogen proteins, particularly in respects to our knowledge in relation to proteomics and understanding new proteins as they're discovered and variants of virus proteins as well, or bacteria. And so the problem really in making these sorts of research tools, particularly antibodies, is that it's a very time-consuming process. It's a process which is extremely expensive, and we've been hearing about the challenges in low middle-income countries in accessing and the equitable approach to accessing these sorts of technologies. And something really needs to change, because at the moment, the cost of making these sorts of new tools is prohibitive. Not only that, there are other challenges as well associated with existing research antibodies. That is, they're quite large. They're about 150 kilodaltons in size, and that means that penetration into tissues is challenging, particularly for intracellular targets. So really, we need a new approach. But if you think about polyclonal reagents, you have variable quality, and of course, making antibodies means that animals are typically required, and there are ethical considerations in regards to that, particularly looking at new approaches that are animal-free. We want to be able to move to that. Antibodies have FC and isotype backgrounds, which can mean challenges in relation to background and non-sort of functional aspects. And they are non-recombinant by default. So really, a radically new approach is needed if we're going to overcome these problems. And that's where Ankyrons come into their own. So Ankyrons are a new generation of technology, which are fully recombinant proteins. They're only 15 kilodaltons in size, so they're a tenth the size of an antibody. And they're highly stable proteins that in their basic form are monomeric in structure. And they're based on ankyrin repeat scaffolds, which are naturally occurring motifs in nature, both prokaryotic and eukaryotic. And we generate these in vitro by ribosome display from a library of more than 1 trillion variants. And that really high diversity library means that we can screen against protein targets incredibly quickly. In a single pass, we can pick out high-affinity binders. So this really changes the whole approach to generating these sorts of reagents, because we can do this in a really cost-effective and high-throughput way. And furthermore, we can, of course, generate multiple clones against targets of interest. So the really exciting thing about Ankyrons is that you can use this technology in all existing applications that you're familiar with. So anything that you're using an antibody for, whether that's immunofluorescence, flow cytometry, ELISAs, immunohistochemistry, you can use Ankyrons in as well. And they have some significant advantages, which I'm going to show you. So the Ankyron advantage is its size. So that means you can penetrate into tissues, particularly intracellular targets, very, very successfully. The other thing about it is that the label on the Ankyron is very close to the point of binding. So that means that the sensitivity of the assay and the resolution, particularly with imaging applications, is second to none. The Ankyrons themselves are generated to high affinity, so we can make Ankyrons even down to sort of low, oops, we can go back one. Down to low affinity, so sort of 0.7 picomolar, for example, against some PDL1 binders that we've generated. The Ankyrons themselves are time-saving, so it means there are no blocking steps required in assays, particularly, like, for example, in flow cytometry, and that means we can actually make a considerable time-saving in lab applications. They're animal-free, as I've mentioned. They're defined recombinant monoclonal sequences, so they're highly consistent, and one of kind of a key thing is that if you've already got your existing antibody panel, these can be seamlessly integrated with those as well. They can be used with existing reagents. You don't just have to have an Ankyron platform. We can also use these and generate them against uniquely distinct sequences. So even if a protein varies by a single amino acid, we can generate Ankyrons against those. And we've done that successfully even against challenging targets like peptide MHC molecules as well. So one key take-home is that Ankyron flexibility is is really exciting because we can label these with pretty much anything. We have a whole variety of different colors. In the most basic format, we provide these with a V5 epitope tag, and so we can use these as a screening platform to pick out the most preferred clone for the application that you're using the Ankyrons in. And we can then also provide those directly labeled with a whole panel of different color options and other label types as well. Also, from a point of view of their format, their most basic format is this monomeric Ankyron, which has high affinity binding. But for certain applications, you may require higher avidity. And that means we can actually make multivalent Ankyrons as well, either through sort of tetramerization or multimeric versions using linkers as well for the future. So this is something which bodes a lot of promise for depending on the specific applications that you're applying them to. We can even make these sort of multi-specific to order, so you can basically plug and play different domains as you require that. So just to kind of give you some examples of some data, this is some immunofluorescence data, not confocal microscopy, but just epifluorescence. And I think you'll agree that the quality of staining you can achieve with these is really second to none. So what you're looking at here in red is an Ankyron stain against an anti-human calreticulin protein. So this associates with the endoplasmic reticulum. The blue is the DAPI stain for the nucleus, and in green is the actin cytoskeleton stain with phalloidin. So you can see really just the quality of stain that one can achieve with this. This is an Ankyron that's been generated against a protein called LRP11, so low-density lipoprotein receptor-related protein 11, not really exactly a short, snappy protein name. But this is a protein that plays a variety of roles in tumor development. And just gives you an idea, because there are no antibodies available against this protein target. And at ProImmune, we can generate these reagents very, very rapidly. So here you can see the Ankyron stain in red, and then the nucleus here in blue. For immunohistochemistry, again, beautiful staining too. Just as an example, we have on the right-hand side here an Ankyron against insulin. And you can see here staining the islets in a human tissue section in the pancreas. So high-affinity Ankyrons can be generated very, very quickly. Particularly, obviously, this is in the context of this audience, really critical for pathogen targets. And the way that this happens is that there is sort of a three-step process. The first thing is what we call Ankyron X. So imagine you're in a situation that you need an Ankyron reagent against a new novel pathogen variant or a protein against a particular pathogen variant. All we need to know is the sequence that you need a protein, an Ankyron against. And the big challenge at the moment is, of course, the antigen availability, where you're going to get this from. You don't need to worry about it. So what we do is we express the protein or the specific domain of that protein in our Ankyron X platform. So we have a variety of different approaches to do that, but basically we make that protein for you. We then screen that protein against our Ankyron Terra library, and then we pick out the Ankyron clones that are binding to that in a very, very quick way. At the moment, this is about sort of a total 12-week process, including the expression of those target proteins. But this is a process which is really accelerating a lot at the moment as well, so that will certainly reduce significantly. And at the moment, we already have a catalogue of thousands and thousands of different target Ankyron clones, covering, I think, 3,600, if I remember rightly, different proteins from a whole variety of different organisms. From a point of view of infectious disease, I've listed these here. Just to give you a bit of a flavour, this doesn't include everything, and probably some of we've obviously heard a lot this morning already about a lot of these different organisms. But the exciting thing is that we have not just Ankyrons against one or two proteins in these, but actually many different proteins within these organisms. If you don't see your organism listed, then obviously you can have a look at our catalogue. But the exciting thing as well is that you can just have a chat with us, and we can actually make Ankyrons for you. So if there's a protein that you want to have a reagent against, you can just have a discussion with us. To take you through some data, we've worked with, for example, the US FDA in a recent publication that came out. This is Ankyrons against spike protein. This is from SARS-CoV-2. In this case, this is some biolayer interferometry data, and what you can see here is the high-affinity binding of the Ankyron against the wild-type Wuhan variant spike protein. And what they did here as a control was actually the Ankyron clones stained against an irrelevant MERS spike protein. So you can see this is a highly specific binding interaction. Interestingly, when they looked at a lot of the different clones, they found they had really high affinity. So, for example, some of the best performing clones in the sort of upper left here are around the sort of 90 picomolar affinity measurements. And the Ankyrons also have functional effects as well. So this is in a neutralization, pseudovirus infection neutralization assay. They were able to demonstrate neutralization effects of the Ankyrons that were generated against that protein. We've worked in the context of vector biology with a group at the Pirbright Institute in the UK. So this is a chap called Kevin Maringer, who's working on Aedes aegypti, and really is looking at the biology and the immune pathways in mosquitoes. And there are no research tools available against some of these proteins, as you can imagine. And this is particularly looking at the NF-kappa B pathway, and there are a number of different proteins, including REL1A, where they needed some reagents against. So it came to us, we made the target protein, we made the Ankyrons against it, we sent them to them, and then they were able for the first time to then have staining reagents to be able to use and understand the biology of this particular model. So that was very exciting, and this paper is in press at the moment. Again, we've been working with a number of different groups. This is the French Institute for Agriculture, INRAE, in France. And this group needed tools to look at Mycobacterium bovis protein MPT63. And in the flow cytometry analysis, you can see an MPT63 negative strain of M. bovis, and on the right, MPT63 positive strain with staining here with the Ankyron in flow cytometry. So in summary, Ankyrons really can accelerate your pathogen research to an extent that's just simply not been possible before. These are very small, so 15 kilodalton binding reagents with high affinity that are cost-effective, rapid, and can be generated in really high throughput. So it allows us to interrogate diseases and species that previously have not been accessible at all. We have more than 16,000 clones targeting 3,200 proteins across more than 140 different species. And if there are particular targets that you're interested in having a chat with us about, please do speak to any of the ProImmune team members here today, because all we need from you is a Uniprot ID, and you can just tell us your protein interests, and we can then express those proteins and put them into our catalogue. There's no cost from a point of view of putting them into the catalogue. All we need to do is just know what you're interested in, and then you can access them from our catalogue. So there's no sort of custom charges, which is a really exciting model and enabling access to those research groups that simply haven't been able to get those before. So thank you very much for your time. I would very much welcome any questions. Thank you. Adam.
Yeah, thank you very much for that. I was wondering, with ribosomal display, is it possible to do non-protein targets, something like LPS or small molecules? Like, can you reappropriate it for that, or does it have to be something you have to make recombinantly in situ?
So at the moment, our strategy has very much been protein-based. So, but the Ankyrons themselves technically can also, there are examples of how those types of molecules have been used against small molecules. But the issue, and I haven't had a chance to talk about it, is really the conformational dependence of the Ankyrons. So they need a globular structure to bind to. So there are challenges with these other types of proteins. Obviously, with LPS, it's depending exactly on what it is, it's obviously a complex mixture of different proteins. So the key thing really for a sensitive specific assay is to have a particular recombinant protein. That's not to say that you couldn't have a number of different Ankyrons against individual proteins, potentially as a combined pool.
Cool, thank you.
Thanks.